Structure of pituitary gland, thyroid, adrenal glands, corpuscles of Stannius and urophysis in fishes. Hormones and their physiological significance. Introduction. In fishes, the endocrine system plays a pivotal role in maintaining internal homeostasis, regulating growth, metabolism, reproduction, osmoregulation, and stress responses. Several endocrine glands are involved in this complex hormonal orchestration. Among these, the pituitary gland, thyroid, adrenal glands, corpuscles of Stannius, and urophysis are the primary centers of hormone production and release. Each gland is anatomically and functionally distinct, secreting specific hormones that influence various physiological processes. This lecture explores the detailed structure, hormonal outputs, and physiological relevance of these glands in fishes. 1. Pituitary gland hypophysis. Structure. The pituitary gland in fishes is a pea-shaped endocrine organ located at the base of the brain, connected via a short stalk called the infundibulum. It is anatomically and functionally divided into three regions: the adenohypophysis, anterior pituitary, neurohypophysis, posterior pituitary, and pars intermedia. Among these, the adenohypophysis is most developed and is further subdivided into regions like rostral pars distalis, proximal pars distalis, and pars intermedia, each harboring specific secretory cells. In teleos fishes, the pituitary is enclosed in a cartilaginous capsule called the sella turcica and is embedded within the floor of the diencephalon. It lacks a portal system in most fishes, unlike mammals, and shows a direct innervation model in neurohypophysis. Hormones secreted. Growth hormone, GH, secreted by somatotropes in the proximal pars distalis, essential for somatic growth, osmoregulation, and metabolism. Thyroid-stimulating hormone, TSH, produced by thyrotropes, regulates thyroid gland activity and influences metamorphosis. Gonadotropins, secreted by gonadotropes, involved in regulation of reproductive cycle, gametogenesis, and steroidogenesis. Prolactin, PRL, plays a key role in osmoregulation, particularly in freshwater adaptation, and in parental behavior in species like tilapia. Adrenocorticotropic hormone, ACTH, stimulates the interrenal tissue, homologous to adrenal cortex, to secrete corticosteroids. Melanocyte-stimulating hormone, MSH, produced by cells of pars intermedia, regulates chromatophores and pigmentation responses. Example: in Carassius auratus, GH levels are correlated with seasonal growth cycles. 2. Thyroid gland. Structure. The thyroid gland in fishes is unique in that it lacks a discrete, encapsulated form. Instead, it consists of scattered follicular epithelial cells located mainly around the ventral aorta and branchial arteries. These follicles are spherical structures filled with colloid, a proteinaceous substance storing the precursor of thyroid hormones. Unlike in mammals, the thyroid in fishes may be unencapsulated, dispersed, and variable in number and size. Hormones secreted. Thyroxine, T4, and triiodothyronine, T3. These iodine-containing hormones regulate metabolic rate, growth, differentiation, and osmoregulation. Both hormones are also involved in larval development and metamorphosis, especially in species like Anguilla and Salmo. They are known to interact with growth hormone and prolactin during osmoregulatory adjustments. Physiological significance. Regulate basal metabolic rate, oxygen consumption, and enzyme synthesis. Influence growth, development, and metamorphosis in larval stages. Control molt and pigmentation in some species. Example: in Channa punctatus, thyroid hormones are essential for metamorphosis. 3. Adrenal glands, interrenal and chromaffin tissue. Structure. Fishes lack a distinct adrenal gland. Instead, the interrenal tissue, equivalent to adrenal cortex, and chromaffin tissue, analogous to adrenal medulla, are separately located. Interrenal tissue is embedded in the anterior part of the kidneys, while chromaffin tissue is often associated with major blood vessels like the posterior cardinal vein. In elasmobranchs, both tissues are closely associated, forming a compact gland. In teleosts, they are more scattered. Hormones secreted. Corticosteroids, cortisol secreted by interrenal cells, regulate energy metabolism, stress responses, and ionic/osmotic balance. Cortisol is the primary stress hormone in fishes and is crucial during salinity adaptation in euryhaline species like Fundulus. Catecholamines, adrenaline and noradrenaline produced by chromaffin cells, mediate fight or flight response, regulate cardiovascular function, and mobilize glucose. Physiological significance. Cortisol plays a major role in osmoregulation, especially in seawater adaptation, and mediates the stress response. Catecholamines, involved in fight or flight responses, increased heart rate, and gill perfusion. Example, in Oncorhynchus micus, rainbow trout, cortisol increases during salinity shifts. 4. Corpuscles of Stannius. Structure. Corpuscles of Stannius are unique to fishes and are small, spherical or oval endocrine bodies located on the surface of the posterior kidney. They are more developed in teleosts and are typically pale in color, surrounded by connective tissue and blood vessels. Histologically, these corpuscles contain Stannius cells or granular cells, which are large, secretory in nature with eosinophilic cytoplasm. Hormones secreted. Stanniocalcin, a glycoprotein hormone responsible for calcium homeostasis. It inhibits intestinal calcium absorption and promotes renal calcium excretion. It acts antagonistically to prolactin in freshwater fishes, maintaining plasma calcium levels within a narrow range. Physiological significance. Primarily regulates calcium and phosphate homeostasis. Inhibits hypercalcemia by reducing intestinal calcium absorption and promoting deposition in bones. Example, in Oreochromis mosambicus, elevated plasma calcium suppresses stanniocalcin production. 5. Urophysis. Structure. Urophysis is an unpaired neuroendocrine organ located at the caudal end of the spinal cord, near the terminal vertebrae. It is a specialized swelling of the spinal cord that contains neurosecretory axons from the nucleus recessus and nucleus of the area postrema in the hindbrain. It is connected to the neurosecretory hypothalamic nuclei and stores, and releases neurohormones into the blood via its rich vascular supply. Histologically, it contains herring bodies, dilated axonal terminals, filled with neurosecretory granules. Hormones secreted. Urotensins, UI and UII peptide hormones that regulate osmoregulation, vascular tone, and blood pressure. UI is involved in sodium retention and cardiovascular regulation. UII has vasoconstrictor properties and modulates renal ion transport. Function. These hormones are crucial for volume and salt regulation, especially during environmental stress or salinity shifts. Physiological significance. Urotensins regulate osmoregulation, cardiovascular function, and smooth muscle contraction. May also play a role in modulating stress responses and neuroendocrine communication. Example, in eels, Anguilla, SPP, urotensin 2 causes vasoconstriction and affects blood pressure. Conclusion. In fishes, endocrine regulation is complex and uniquely adapted to aquatic life. The pituitary gland coordinates a multitude of physiological processes through trophic hormones. The thyroid gland regulates development and metabolism, while interrenal and chromaffin tissues provide stress and metabolic control. The corpuscles of Stannius and urophysis, being unique to fishes, reflect the evolutionary specialization in calcium balance and osmoregulation. Together, these glands ensure homeostasis, development, and adaptability in a range of aquatic environments, providing critical insight for fields like aquaculture, developmental biology, and comparative endocrinology.